Hello, everyone, and welcome to today's live broadcast. Lactate-driven metabolic adaptation modulates breast cancer progression, presented by Jennifer Molina, PhD. We're excited to bring you this educational web seminar presented by LabRoots and sponsored by Agilent. Agilent is a leader in life sciences, clinical research, diagnostics, and applied chemical markets. With over 11,000 employees worldwide, the company provides laboratories worldwide with instruments, services, consumables, applications, and expertise, enabling customers to gain the insights they seek. Agilent's expertise and trusted collaboration provide the highest confidence in its solutions. Agilent focuses its expertise on 6 key markets where the company helps its customers achieve their goals: food, environmental and forensics, pharmaceutical, diagnostics, chemical and energy, and clinical research. For more information, please visit www.agilent.com. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. This presentation has been approved for continuing educational credits. If you want to obtain the credits, please click on the Get Your Free CME CE Credits button located in the lower left of your screen. This will take you to the continuing education page where you can select speaker, click on the CE/CME button, and it will take you through the process to obtain your certification. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window. Type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please note that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. And finally, if you have any trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know you're having a problem. Thank you. Now I'd like to introduce Jennifer Molina, PhD, Institute Research Scientist at the Institute for Applied Cancer Science at MD Anderson Cancer Center. Dr. Molina has a PhD in biomedical science from the University of Texas Graduate School of Biomedical Science and research experience in the fields of cancer biology, biochemistry, cell signaling, and drug discovery. Dr. Molina joined the Institute for Applied Cancer Science, IACS, in 2015 following a postdoctoral fellowship in the Department of Systems Biology at MD Anderson Cancer Center, where she focused on uncovering mechanisms of metabolic adaptation in breast cancer for the discovery of potential therapeutic metabolic targets. Dr. Molina is an Institute Research Scientist in the Biology Division at IACS. Please join me in welcoming Dr. Molina. I will now turn the presentation over to her.
Thank you very much, Judy. I hope everyone can hear me, and I want to thank Agilent Technologies and LabRoots for the opportunity to present my work. As Judy mentioned, I finished my postdoctoral training in systems biology in the laboratory of Gordon Mills, and the work I'm presenting today was all done during that time. And about 3 months ago, I joined IACS, also at MD Anderson, and I'm working on novel metabolic targets in cancer. So for today, the title of the talk is Lactate-Driven Metabolic Adaptation Modulates Breast Cancer Progression. Breast cancer is a devastating disease that affects many people every year. In 2014 alone, there were over 200,000 new cases in females and over 2,000 cases in male. It is a very heterogeneous disease, and extensive molecular profiling has uncovered that there are several subtypes of breast cancer that are characterized by key genetic mutations and amplifications, and they are highlighted on the left. The subtypes are luminal, which is characterized by increased expression of the estrogen and progesterone receptors; HER2 amplified, which is characterized by amplification in the ERBB2 gene; There is also a small subtype called the claudin low, which is more stem-like in behavior. And then there is the infamous triple-negative breast cancer subtype, which has low estrogen and progesterone receptors and high expression of other markers like the EGF receptor and so on. And it can also have increased HER2 expression. These subtypes also represent different outcomes in patients, and you can see this on the right-hand side. And this graph here expresses overall survival in patients. And as you can see, the luminal subtype in blue has the best prognosis as compared to the rest of them, where triple-negative subtype and HER2 subtypes have the worst outcome in patients. Along with the subtypes that were uncovered in patient samples, analysis of 60 different breast cancer cell lines, and this has been done by multiple groups all over the world, uncovered that the cell lines retain the same subtypes as the patient samples do. So this makes them valuable tools to study breast cancer biology. And that's exactly what we have done. Because we know that breast cancer is a very heterogeneous disease genetically and epigenetically, we also wanted to know if this heterogeneity is found as far as metabolism and how cells metabolize the major nutrients, glucose and glutamine. And in this graph, what I wanted to show was the workflow of what we typically do to begin examining like basal metabolism in breast cancer cells. First of all, the two major nutrients in cancers are glucose and glutamine. Glucose can be metabolized to make pyruvate, and then it has multiple fates, one which is flux through the TCA cycle and electron transport chain, and the other one is conversion to lactate, which is called the Warburg effect or aerobic glycolysis in the presence of oxygen. The other nutrient is glutamine, which can be converted to glutamate and either also feed the TCA cycle or be secreted as glutamate. What we usually perform to test differences in breast cancer cells is to take fresh medium and compare it to spent medium after a certain time points. And what we can calculate, and this was done by using the YSI metabolic analyzer, full protocols are found in the citation seen here. We can see consumption of glucose over time and production of lactate over time, and we can calculate glycolytic ratios from this. And we can also examine glutamine consumption and glutamate secretion into the media. And I'll be talking about this further once we get more into the data. Another method that we typically use to analyze basal metabolic rates in breast cancer cells is using the EXEV analyzer by Seahorse Biosciences. This methodology allows us to look at the oxygen consumption rates as well as the extracellular acidification rates, which is typically a measure of lactate production in cells in real time. In this particular example, we use the mitochondrial stress test, which is one of the methods that can be used in this assay. The assay allows us first to interrogate basal metabolic rates. So how much oxygen is consumed by cells and how much lactate is produced in real time. And then it allows you to inject different inhibitors, up to 4, and see what happens to the OCAR and ECAR in real time. So in this case, we used 4 different mitochondrial inhibitors. First, we used oligomycin, which is an ATP synthase inhibitor, and you can see a drop in the OCAR. Next was an FCCP, which is an uncoupler that allows electron flow, and it shows the maximal respiratory capacity, so how much cells can compensate during stress. And then finally, a complex 1 and 3 inhibitors, antimycin and rotenone, which completely kill OCAR, and then it allows you to calculate the spare capacity. So here, I'm just illustrating 2 different cell lines. As you can see, the basal metabolic rates are different in the 2, and you can also get differences in maximal respiratory capacity. And these are also, you're able to correlate with responses in the extracellular acidification rates. And finally, that allows you to calculate the amount of oxygen that is consumed to generate ATP for ATP production. So we apply this type of exercise in a panel of breast cancer cells. And to our surprise, we found that if we looked at compared basal or triple negative, which are in black, and we compare them to the luminal subtype, which are here in red, and this is OCAR ATP over ECAR, the ratio, we found that the luminal subtypes tended to be more oxidative, and the basal subtypes tended to be more glycolytic. Next, we correlated these differences looking at gene expression in the different subtypes, and we found that actually a lot of the major metabolic enzymes were differentially expressed, and there was a dichotomous expression, and a lot of these enzymes are responsible for different types of metabolism. So some of the most significantly differentially expressed genes are involved in lactate transport and metabolism. So the #1 dichotomous expressed gene in breast cancer that we were able to identify is LDHB, the lactate dehydrogenase B isoform. This is the enzyme that can convert pyruvate to lactate. In addition to that, the 2 transporters of lactate, which are here, SLC16A1 and 3, or MCT1, MCT4, were also dichotomously expressed. So some cells have very high levels, some cells had very low levels of these genes, and that also translated to protein levels. And finally, FBP1. And as you can see, this differential expression is significant because here are the estrogen receptor and the HER2 gene, which are used to subtype and clinically select the treatment in patients. So if we compare the differential expression of some of these metabolic genes as compared to what is used diagnostically, it is significant. So this sparked our interest in lactate biology in breast cancer, and we continued looking at what can be going on with lactate biology. When we look at patient data, on the left, we can see this is survival of patients within the triple negative subtype of breast cancer that have high levels of MCT1, the lactate transporter. We see that survival is significantly lower than patients that have low lactate transporters. And this also translated to the expression levels of MCT1. And this data was analyzed from the TCGA database cited here. Other studies have also associated that high lactate was found to be high lactate in patients in clinical samples was associated with chemotherapy and radiotherapy resistance, immune system evasion, high angiogenesis in tumors, and increased metastasis. And on the right is an example here where samples were examined from human cervix cervical cancer, and those tumors that had high lactate in the microenvironment had worse survival. In addition to that, recent publications have shown that there is heterogeneity in the preference of nutrients that different cells that make up tumors consume. Some tumor cells and areas are very glycolytic, while others prefer glutamine consumption or lipid consumption. One particular study showed that there is an inverse correlation in the level of lactate in tumors. Where lactate is high in tumors, glucose tended to be lower, and vice versa. And that also correlated with ATP levels. So this and other studies influenced our curiosity to study what lactate can provide cells during nutrient stress when glucose or glutamine levels are low. And perhaps that could play a role in the survival. So this was our original hypothesis, that when breast cancer cells have plenty of glucose, they can metabolize glucose to make pyruvate and then eventually to make lactate. Lactate accumulates in the microenvironment, as has been shown by many different studies. And the cells can also metabolize pyruvate from glucose through the TCA cycle to make plenty of ATP. However, there are many studies showing that cancer cells tend to have a preference for glycolysis or lactate production, which makes very little ATP as compared to full oxidation through the TCA cycle. So we wonder, why is this? It seems like a wasteful mechanism that throws out perfectly good carbons into the microenvironment. So we hypothesized that lactate accumulates, but in areas where glucose is low as compared to areas where glucose is high, both glucose and glutamine are low, cells are under nutrient stress. And perhaps they could utilize the lactate that accumulates in the microenvironment, which is plentiful and can go up to about 10 millimolar as compared to physiological, which is only 1 millimolar, and feed cells during times of nutrient stress, so cells could make whatever they need to survive. Okay, so we wondered if this was happening and what pathways could be utilized by lactate metabolism under nutrient stress conditions. So to interrogate this in terms of a screen in many different breast cancer cell lines, we chose just very simple conditions to compare and to look at survival. So we chose a physiological media condition, which contained physiologically relevant levels of glucose, glutamine, or lactate, which is 5.5 and 1 millimolar respectively, compared it to a base medium without the 3 nutrients, which we term nutrient-free, and it's a negative control. And then we compared that to a high lactate condition of 10 millimolar lactate, which is typically found in breast tumors. And we analyzed induction of apoptosis in the different conditions after treatment. And then we did a number of other assays, and I'll go over all this data subsequently. So during our screen, we looked at percent survival in physiological, and as you can see, we had 100% survival overall. While in nutrient-free conditions, we had 20 out of 24 breast cancer cells that died, which was expected. And then when we added high lactate to that nutrient-free media, we found that most of the cell lines received some significant benefit from having lactate in the media. Basically, there was a delay in the apoptosis of these cells. But most strikingly was that 7 out of those 24 cell lines, and these are highlighted in red, were really lactate-sufficient cells. They survived very efficiently in high lactate alone. And if we looked at the subtypes of these cells, we really didn't see, we saw an enrichment in the basal or triple negative subtype as far as surviving, but we also saw a few that survived in the luminal subtype and one HER2 positive. So for the more mechanistic studies, we chose two cell lines to compare. And we chose MCF10A cells, which are derived from normal breast epithelium. And we also chose the HCC 1937 cells, which are basal triple negative cells, to do further studies with. So this is a measure of percent viable cells by exclusion of apoptotic cells in the presence of either physiological media, which of course we see survival over 4 days, nutrient-free, we see rapid cell death, and high lactate, there is a significant maintenance of survival even after 4 days in high lactate media. And if we compare that to the MCF10A cells, we see that there is a significant difference in the number of cells surviving. This was also consistent with the maintenance of morphology and size in HCC 1937 cells, which are the lactate-sufficient cells versus the normal cells. The next kind of obvious place to look was energy and redox balance. So we started looking at AMP to ATP ratios in both conditions and found that there was a maintenance of ATP in both the HCC 1937 cells as well as the MCF10A cells. So this told us that lactate is providing ATP even in the less sufficient lactate sufficient cells. So ATP maintenance is not part of the full story here, but it certainly plays a role. Next, we looked at NAD to NADH ratios and found the same maintenance of NADH in both the lactate sufficient and the lactate insufficient cells, which tells us that, again, this is part of the story, but not the full mechanism. And then finally, we looked at oxygen consumption by the Seahorse XF analyzer and found significant increase, very significant increases in the oxygen consumption when lactate is present in the HCC 1937 cells, and only a minor oxygen consumption increase in the other cell line. So this told us that perhaps we should be looking at lactate being converted to pyruvate, which can be metabolized into the TCA cycle for NADH production, and then taken into the electron transport chain for ATP production. Finally, by using the YSI analyzer, as I was previously talking about, analyzing lactate levels in the media, we confirmed that in fact, when lactate is present at high levels, it is consumed by the cells in both the MCF10A and the HCC 1937 cells. So to summarize part one, we found that cells deplete exogenous lactate from the media. This promoted survival, helped to maintain ATP and ADH levels, and increase the oxygen consumption. So next we wanted to know the mechanism of what else is helping cells to survive when lactate is present. So for that we used three different methods. First, we used labeled carbon-13 labeled lactate, fully labeled lactate, to do mass spectrometry analysis as well as NMR analysis. But then our main experiment was really to look in an unbiased way to do metabolomics analysis in the presence of lactate in the HCC 1937 cells to see what could be happening there. So this is the workflow that we used to analyze our cells. So after sample preparation, we compared high lactate cells grown in high lactate media versus nutrient stress condition. We did the examination by LCMS analysis using the Agilent C550 iFunnel QTOF LCMS with a helic column, helic chromatography. The chromatogram alignment was done by mass feature extraction using the Mass Hunter ProFinder software. Statistical analysis was done to identify the most significantly changed metabolites, and then the Mass Profiler professional software was used to identify what these metabolites are. And this work was done in collaboration with Agilent Thought's leader scientists, as well as Pietro Morlacchi here at Ajax, MD Anderson. Here are the results of our unbiased screen. 280 features were extracted with ProFinder. 86 were significantly different between the two conditions, and about 50% were annotated with the Metlin database. And as you can see, the overall heat map, where nutrient-free is on the left and lactate is on the right, you can see that there are significant differences in several metabolites. And I'll get into the specifics here. So overall, when lactate was present, we saw that several TCA cycle components were increased, and they are here: oxaloacetate, cisaconate, succinate, fumarate, and malate were increased as compared to nutrient-free media. Several amino acids were also increased, including glutamine and glutamate. And several nucleotides were increased, as well as a few glycolysis, as well as pyruvate, pyruvate. So this gave us an idea of where to look specifically within the different central carbon metabolism pathways. So we moved forward to do NMR analysis with fully labeled lactate to see where specifically the carbons from lactate were found. And here is the overall picture. So on the top is the HCC 1937 cells, and on the bottom we have the normal NCF10A cells. And the main difference here is that the HCC 1937 cells were really able to produce a lot of different metabolites as compared to the MCF10A cells. So the main amino acids that were found to be enriched with lactate carbons are glutamine, glutamate, alanine, aspartate, and glycine. These were all made in the lactate-sufficient cells, while in the lactate-insufficient cells, the main amino acids were only glutamine and glutamate. In addition to that, we saw that there is a big enrichment and flow of lactate carbons through many different cycles in the TCA cycle, which produced specific label patterns in the glutamine and glutamate. So after round 1, we see this pattern, and after several rounds, there's a saturation of the signal. Another very important point that we observed was that oxaloacetate or aspartate, which comes from oxaloacetate, had different signaling carbon patterns. So that told us that one of the versions of aspartate is coming from oxaloacetate that comes from the TCA cycle, but another one is actually coming from a direct reaction coming from pyruvate to oxaloacetate conversion, and this is done by pyruvate carboxylase enzyme. Finally, when we looked at the patterns of labeling in glycine, that also correlated to That gave us a hint that perhaps we should be looking at gluconeogenesis or reverse glycolysis, because glycine comes from the serine pathway arm of glycolysis. And in this case, we don't have any glucose in the environment, in the media. But I'll get into the specifics of that a bit later. Okay, so because we know that robust electron transport chain oxidative phosphorylation is also related to production of high levels of reactive oxygen species, and that's toxic to cells. We also looked at the major antioxidant in cells, which is glutathione. Since we observed that cells can make glutamate and glycine from lactate, and these two are components of glutathione and are used in its de novo synthesis of glutathione, we thought, okay, maybe the cells can take these amino acids they make from lactate to make glutathione to relieve some of reactive oxygen species that they are producing from OXPHOS. So we looked at this in many different ways. First, we looked at levels of hydrogen peroxide, and we found that lactate maintained lower levels of hydrogen peroxide in HCC 1937 cells, while in MCF10A cells, first of all, levels were higher to begin with, but they were not maintained or decreased in the presence of lactate. Next, we looked at NADPH production, which is also necessary to deplete ROS from cells, and we found that NADPH is maintained in the presence of lactate in HCC 1937 cells, while it is lost in MCF10A cells, even when lactate is present. And then finally, we looked at levels of reduced glutathione by using carbon-13 labeled lactate again in HCC 1937 cells, and found that there is enrichment of carbons coming from lactate in reduced glutathione. This tells us that cells were utilizing the carbons from lactate to make glutamate and glycine that can become de novo glutathione. And that was, again, helping for survival. And as a proof of concept, we used BSO, which is an inhibitor of the synthesis of glutathione. And every time that we did this in many different cells, when we treated cells with BSO, we saw a significant decrease in the survival of cells in lactate. And this was rescued by the addition of glutathione. So together, all this data tells us that there is biosynthesis of glutathione from lactate metabolism, and that is required as part of the survival under nutrient stress, and that increased reducing power is associated with lower survival. And that is the mechanism, one of the mechanisms we found that lactate is helping with during nutrient stress. Okay, and so next we wanted to interrogate how cells could possibly be making serine or glycine when they only have lactate present. So in this pathway, we can see glycolysis, and at the 3PG branch is where the serine producing pathway is found, and that leads to also glycine production. So we hypothesized that cells could perhaps be taking lactate, metabolizing it through the TCA cycle to make oxaloacetate and aspartate, and then that aspartate could in turn be reconverted to oxaloacetate in the cytoplasm and be converted to phosphoenol pyruvate, which is the second step of reverse glycolysis, and the cells could carry on up the pathway backwards to make 3-PG, and that could lead to glycine production. The next question we also had was if, in fact, cells were able to do this reverse glycolysis, how far up the pathway they could be going, if perhaps they were able to go all the way up to the pentose phosphate pathway, which is a major production of NADPH, which is needed to maintain reduced levels of glutathione to combat toxicity of reactive oxygen species, as well as eventual production of nucleotides. So the way we looked at this was to interrogate the first two steps in reverse glycolysis. First, we knocked down pyruvate carboxylase, which is the converts pyruvate to oxaloacetate. And after knockdown of PC, we observed a significant decrease in the survival of HCC 1937 cells. But this actually was rescued by addition of glutamine to lactate. And that makes sense because glutamine can be converted to glutamate and enter the TCA cycle as alpha-ketoglutarate and can provide the oxaloacetate needed to circumvent this problem. So cells could actually make aspartate from that oxaloacetate derived from the glutamine. So that rescued aligns with what we know. Next, we also looked at the second step of reverse glycolysis, which is conversion of oxaloacetate to phosphoenolpyruvate. And we did this by treating cells with the drug troglitazone, and found that there was a very, very robust decrease in the survival of cells in the presence of lactate when PCK1 is a PCK1 inhibitor. When we treat the cells with this, however, this time, as we would expect, we didn't see any rescue with the addition of either glutamine or glutamate. And that also made sense with what we know about these pathways, because cells, even if they can undergo anaplerosis from glutamine and glutamate to make aspartate and oxaloacetate, they would not be able to convert it to phosphoenolpyruvate. So this was a first hint that reverse glycolysis may be involved in the metabolism of lactate. And in fact, we know from normal physiology that in the liver there is metabolism of lactate to make glucose. So the next step here was to really look at some of the components of this pathway of reverse glycolysis and pentose phosphate pathway and serine pathway by doing mass spectrometry with carbon-13 labeled lactate. And we found that in all cases, we found that carbon-13, carbons coming from lactate were found in serine, first of all, so that confirms why glycine was produced. Also, we found label in G6P, which tells us that cells were able to metabolize carbons from lactate all the way up to the pentose phosphate pathway. In addition to that, we also found label in several different nucleotides, like or first backbones of nucleotides, R5P, IMP, ADP, ATP, NAD, and NADP. These are all backbones or purine nucleotides. And then when we inhibited G6PD which is the first step in the pentose phosphate pathway, it's an inhibitor of G6PD, DHEA, we saw a robust decrease in the survival of cells grown in lactate. So it tells us that part of the mechanism which is required for survival under under lactate metabolism is the ability for cells to metabolize carbons through the pentose phosphate pathway. So in order to see if we could apply this mechanism as part of what we know in breast cancer, we chose to analyze combinations with two different inhibitors. First, we thought, okay, we could induce nutrient stress by treating cells with 2-deoxyglucose, which is a glycolysis inhibitor. It competes with normal glucose, and cells retain, they cannot undergo glycolysis past the first step of glycolysis. So in the first scenario that we could think of, if we would treat cells with 2DG alone, and they are lactate-sufficient cells, they should be able to compensate by metabolizing lactate with the pathways that I just described and undergo reverse glycolysis to run the pentose phosphate or the serine glycine producing pathways and TCA cycle. So this should not be, in theory, very toxic to cells. On the other hand, if we treat with troglycine alone, and this is, of course, under physiological conditions when cells have glucose, the cells are not dependent on reverse glycolysis in this case. So inhibiting PCK1 should not be very toxic to cells either. But if we combine the two, so we make the cells, we can make the cells dependent on lactate metabolism after 2DG treatment, and then blocking PCK would prevent them from metabolizing lactate through the reverse glycolysis, and in theory there should be an additive effect between the two, between the combination. So we started this with just cell lines, and this is a HCC 1937 cell. Looking at survival first, under high levels of glucose, so physiological levels of glucose, which is 5 millimolar. And our reasoning behind this was that normal cells have physiological levels of media as compared to tumors that are where lactate is high, glucose would be low. So this is why we chose this condition. So under physiological conditions, a normal cell would not suffer the toxicity levels achieved with the combination of these two compounds. Under nutrient stress conditions, however, when there is no glucose, both the combination and troglycine alone were able to really decrease viability substantially. And then in areas in a tumor where glucose is low and lactate is high, treatment with the single agents alone did not have any effect. However, the combination was very effective at decreasing, at killing cells. And we were able to repeat this in several other lines with the same additive effect between induction of nutrient stress by inhibition of glycolysis and then inhibition of PCK by treatment with troglycine. And we did this in a total of 4 different lactate-sufficient lines, which are here. Also MDA-MB468, which is a triple negative line, SKBR3, which is a HER2 amplified line, and CAMA1, which is a luminal line. There were a few differences in the potency of troglycine in a few lines, and that correlated with the actual mRNA levels of PCK1. So that could be a hint that in that particular case, you would need to hit PCK1 harder. And then this was the most interesting part. After this experiment was done, I took the remaining cells in the dish and just removed the drug and replenished fresh, full, rich cell culture media and waited to see what cells came back. And in all cases, the combination between 2DG and triglyceride did not have any cells coming back and restarting in proliferation. So there were no surviving cells after drug removal, while the single treatments did not have the same effect. And then finally, we analyzed these effects in vivo in an orthotopic xenograft model with the HCC 1937 cells. And as you can see, there is an additive effect in tumor growth over time. Although we didn't quite see a regression, we saw a cytostatic effect or maintenance of a low tumor size as compared to the single treatment alone or vehicle. And the main point was that whatever grew, the tumor sizes remained quite small as compared to the single treated tumors. This could be because there is, I think this speaks to the fact that if we are really thinking that this combination, especially targeting PCK for lactate-dependent tumors, we would benefit from using a more potent inhibitor of PCK. So this is just to summarize the mechanisms that so far we've uncovered and others as well. Lactate is converted to pyruvate and metabolized in cells for continuous rounds of the TCA cycle. OXPHOS generates both ATP and NADH as well as ROS, reactive oxygen species. These are toxic to cells, so that probably signals cells to start producing some amino acids. And what we found so far is that cells made glutamate, glutamine aspartate, serine, glycine, and alanine, all from lactate. And with those amino acids, they were able to synthesize glutathione, maintain ROS levels low, and by employing reverse glycolysis, they made additional amino acids, some which are required to make glutathione. They increase or maintain levels of NADPH, and they were able to make purine nucleotides, all for survival. So here are some of the lessons learned and that we can apply. Lactate, in this case, based on these mechanisms, may present a roadblock for targeting metabolic enzymes by enabling resistance. And I say this because cells were able to make many different amino acids and nucleotides from lactate. So targeting certain metabolic enzymes in the presence of high lactate might compensate by actually making the products or metabolites that you are trying to inhibit. One of which can be production of glutamate, which right now is under study by many different groups. So a potential application or intervention strategy for lactate biology could be that induction of nutrient stress, perhaps by glycolysis inhibition, could be an arena in which you create lactate-dependent cell populations that emerge and that you can target by inhibiting one of the enzymes that cells become dependent on when they are using lactate, like PCK. So it would be interesting to work with better compounds of PCK to understand if this is feasible or not. And with that, I'd just like to acknowledge everybody that worked on this team. So like I said, this work was done in the laboratory of Gordon Mills at MD Anderson Cancer Center, Department of Systems Biology. These were all my collaborators in the lab listed here. I'd also like to acknowledge the CCSG characterized cell line core, All the mass spectrometry and NMR work was done by Pietro Morlacchi in collaboration with Agilent thought leader scientists. And also the NMR was done in collaboration with Kumar Kaularachi. And I also worked with Leslie Silva for some mass spectrometry analysis. So thank you very much for your attention. And I guess now we can move to some questions.
Yes, thank you, Dr. Molina, for that informative presentation. It is time for Q&A. So if you have a question you'd like to ask Dr. Molina, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many questions as we have time for. So let's get started. Our first question is, how general do you think this mechanism of lactate metabolism is across various cancer types?
Yes. So I think it can be very applicable because, as we know, most cancers, especially solid cancers, are now being detected by FDG-PET scan, and that is essentially a measure of glycolysis. So glycolytic tumors light up, and that's how they are detected. And you can, we know that glycolysis means lactate production. However, the actual application of targeting lactate is not going to be as a single agent treatment. I think it should be explored with greater detail in the context of nutrient stress. So if you were to target lactate alone, you're going to hit a subpopulation of cells in tumors that are under nutrient stress. Characterizing what population that is would be interesting, but then also thinking about combinations with maybe standard of care or other methodologies like targeted therapies would be interesting to explore. So I think that there's a lot of work to be done. There have been some hints about targeting the import of lactate into cells. There's work by several groups showing that for some cancer types, it can have some effect, but I think it's still too early to tell how general this can be. And
why the focus on measuring metabolites?
Can you repeat that? Why the?
Okay, I'll just repeat that question. Why the focus on measuring metabolites?
Yeah, so the focus on measuring metabolites for this particular case was very important because, for example, if we had only looked at protein expression or gene expression, that could have told us some changes in in the levels of different enzymes that might be changing. For example, if originally we looked at mRNA levels of the different enzymes, but that doesn't really tell us what is going on in the cells. I think interrogating the levels of metabolites across different conditions can really tell us what the cells are doing as far as metabolizing the major nutrients like glucose and glutamine. So that we can assess much better what, in terms of cancer and targeting, what the pathways that are being utilized by different cancer types. Knowing the levels of the metabolites is essential in really figuring out how the cells are surviving and proliferating and what they're using the different sources to make. So if we have found that lactate was not just used to make ATP, I think that was just part of the question. If we had not looked at more than that, we would never have uncovered all the other amino acids and the nucleotides.
What about IMP?
So IMP, we found lactate carbons in our mass spectrometry labeled lactate experiment. We did find that IMP was labeled from lactate, and we think that it was from the flux through the pentose phosphate pathway. We found R5P and IMP labeled, and we also found other nucleotides. So I think I summarized NAD, NADP, ATP, ADP, as well as R5P. And when we inhibited the pentose phosphate pathway by treatment with DHEA, we saw a decrease in the survival of cells. I did not try to rescue the effects of inhibition with any nucleotides, but that's something that I could try to do. I know that there were recent publications, I think within the last month, that showed the importance of OXPHOS and the electron transport chain in the production of nucleotides via the production of aspartate. So I think that that's certainly a mechanism that's tying into this story, that lactate provides the aspartate that cells need to make nucleotides, and the electron transport chain and OXPHOS is certainly something that lactate is contributing to for that purpose.
What genomic mutations or gene expression changes are associated with lactate sufficiency?
Yes, so as far as genomic mutations, I'm not really sure, but as far as gene expression levels, I would say that the cells that tend to have higher gene expression levels of the lactate transporters like MCT1, MCT4, as well as higher levels of gene expression levels of the lactate dehydrogenase enzymes LDH-A and B are found to be lactate sufficient. Furthermore, in our project, we also looked at levels of the enzymes involved in gluconeogenesis. So FBP1 was associated with lactate sufficiency, as well as PCK and PC, levels of pyruvate carboxylase and phosphoenol pyruvate carboxykinase. All of those were associated with sufficiency with lactate.
The next one is a two-parter. Have you looked at cancer cell lines of other types? And what about prostate cancer?
So the next question was, if I've looked at cancer cell lines of other types, what about prostate cancer? And I think I have not personally done this work, but I think there is work that has been done in prostate cancer where they have found that lactate maintained viability and survival in prostate cancer cells.
In the slide in which you show the increase of different compounds in cells incubated in the absence of lactate, you didn't comment what happens to nucleotides. Can you give us some information about, in particular, regarding IMP?
Okay, next question. I think I have a partial question here, but it's talking about the increase of different compounds in cells incubated in the presence of lactate. But I'm sorry, I'm not. Oh, in the presence of lactate. I didn't comment on what happens to the nucleotides. Okay. I think it's talking again about IMP. I think I answered all I know about IMP at this point was that we found carbons coming from lactate enriched in IMP. The percentage was not huge, but it was there.
Targeting lactate metabolism in breast cancer could alter calcium levels and release from the endoplasmic reticulum. I'm not sure if that's a question or a comment, so if you have a thought to share, please do.
Okay, so the question is, targeting lactate metabolism in breast cancer could alter calcium levels and release from the ER. I haven't looked at calcium levels. I'll tell you that the death that I am seeing is apoptosis. I didn't, and the way I know that is because all my viability assays that I show to show percent survival are labeling with DIOC6 as well as propydium iodide. However, I didn't dive into all the different types of apoptosis, but I know for sure that it is apoptosis. But I have not looked at calcium levels.
We're almost out of time, so this will have to be our last question. Is there any known correlation between psychological stress and tissue cellular stress? And second part, reverse glycolysis is glutathione production? Glutathione production.
Okay, so for the first part of the question is asking about a correlation between psychological stress and tissue cellular stress. I am not an expert in psychological stress, so I'm afraid I cannot comment on that. I'm wondering if that was physiological stress instead of psychological stress. I'm not sure. If we're talking about physiological stress as far as levels of decreased glucose or glutamine, that should have an impact on the metabolism, cause nutrient stress and therefore cellular stress. And lactate was we and others are showing that lactate is can be a compensatory mechanism because it's allowing cells to make some of the biomass molecules they need to survive. And then the second part of the question is reverse glycolysis glutathione production? So reverse glycolysis is essentially gluconeogenesis, is basically when cells flux through the glycolysis pathway backwards. And in, for example, liver cells, they do this, and the glucose they make can also be stored as glycogen. In our case, we didn't find flux all the way to regular glucose, but we did find that cells were going through glycolysis to produce first serine and then glycine. Glycine is required for de novo synthesis of glutathione. Glutathione is a tripeptide that contains glutamate, cysteine, and glycine. So lactate was able to provide carbons to make 2 out of the 3 amino acids that make up glutathione. And when we looked at labeled carbons coming from lactate in glutathione, we found a significant percent of glutathione was labeled from lactate carbons. So it contributed for that production of glutathione.
I would like to once again thank Dr. Molina for her presentation. Do you have any final comments, Dr. Molina?
I just want to thank everyone I just want to thank everyone for your attention, for all your great questions. If you want to communicate with me in the future, I am on LinkedIn, so please find me. I'd be happy to answer all the questions you have and maybe set up some collaborations. And maybe I'll have some questions myself for you in all of your research that you do. So thank you so much. This was a wonderful experience. Thank you to Agilent Technologies and LabRoots for this opportunity.
Thank you. Attendees should check back later for a PDF of this presentation. And literature on cancer metabolomics is available at the Agilent booth, and you may go there to further speak with experts. Head over to the Agilent booth. Just a reminder, if you wish to obtain the credits for today's session, please click on the Get Your Free CME CE Credits button located in the lower left of your screen. This will take you to the continuing education page where you can select a speaker, click on the CE CME button, and be guided through the process to obtain your certification. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing for 6 months from today's live event. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. See you again soon. Goodbye.